Welcome to Mootechie, Microbial Commensalism and Colonization, Understanding Host Microbe Relationships. In this session, we will understand this topic step by step. Now, let us begin by understanding microbial relationships. First, let us look at mutualism. In mutualism, both the host and the microbe benefit from each other. This is a positive interaction where both sides gain something useful. Next, we have commensalism, which is our main focus. Here, the microbe benefits but the host remains unaffected. The host is neither harmed nor helped. The host simply tolerates the presence of the microbe. Another important relationship is parasitism. In this case, the microbe benefits but the host is harmed. This often leads to disease conditions. Finally, there is neutralism. In neutralism, neither the host nor the microbe affects each other in any significant way. Now, let us clearly understand what is commensalism. First, let us look at the commensal organism. The commensal benefits by getting nutrition, shelter, and protection from the host environment. Next, let us consider the host. The host remains unaffected. There is no harm done, no benefit received, and the host simply tolerates the presence of the microbe. So, commensalism can be defined as an association where the microbe benefits while the host remains neutral. Now, let us see some examples of microbial commensalism. First, on human skin, we commonly find Staphylococcus epidermidis. It lives on the skin surface and helps prevent colonization by harmful pathogens. Next, in the intestinal tract, bacteroids species are present. They help break down complex molecules and support digestion. Another example is in the oral cavity where Streptococcus viridans is found. These bacteria are abundant in the mouth and are mostly harmless. Finally, in the rumen of cattle, cellulolytic bacteria are present. They help in digesting cellulose, which is essential for ruminant animals. Now, let us understand the concept of normal microbial flora in different parts of the human body. First, the oral and respiratory regions. These areas have a high population density due to moist conditions. Common microbes include Streptococcus viridans and Neisseria. Next, the skin surface. The skin is dry, salty, and acidic, acting as the first line of defense. Staphylococcus epidermidis and Propionibacterium are commonly found here. Moving on to the gastrointestinal tract, this is the largest microbial community in the body and plays a major role in digestion and immunity. Bacteroids and E. coli are important members. Finally, the urogenital tract. Here, microbes like Lactobacillus maintain acidic pH, helping prevent infections. Now, let us understand the benefits of commensal microbes to the host. First, protection. Commensals occupy space and nutrients, preventing pathogens from adhering and growing. They may also produce antimicrobial substances. Next, nutrition. Some commensals synthesize vitamins like vitamin K and B12. They also help digest complex carbohydrates and produce short-chain fatty acids. Another important benefit is immune balance. Commensal microbes train the immune system to differentiate between self and non-self and help maintain a balanced immune response. Now, let us understand the concept of microbial colonization. First, adhesion. Microbes attach to specific receptors on the host surface using structures like pili or fimbriae. Next comes growth. Once attachment is successful, the microbes multiply using locally available nutrients. Finally, persistence. The microbes form stable colonies or biofilms that help them resist removal and survive for long periods. Now, let us look at the sites of microbial colonization in the human body. First, the respiratory tract. Mucosal surfaces trap microbes and colonization is heavier in the upper respiratory tract. Next, the skin surface. It is dry, salty, and acidic, acting as a physical barrier. Microbial distribution varies by skin region. Moving to the gastrointestinal tract, this is the largest microbial community in the body and plays a major role in digestion and immunity. Next, the urogenital tract. Here, microbes like lactobacillus maintain acidic pH, helping prevent infections. Now, let us understand the factors affecting microbial colonization. First, host factors. The immune system, especially IgA antibodies, prevents overgrowth. Physical conditions like pH, moisture, and temperature also affect survival. Availability of surface receptors influences attachment. Next, microbial factors. Microbes use adhesins like pili for firm attachment. They form biofilms for protection and efficiently utilize local nutrients for growth. Now, let us differentiate between colonization and infection. First, colonization. Here, microbes establish a stable presence without tissue damage. There are no symptoms and the host remains healthy. Next, infection. In infection, microbes invade tissues, cause damage, and produce symptoms like fever, pain, and inflammation. Medical intervention is usually required. Now, let us see the role of commensals in preventing infection. First, no space. Commensals block attachment sites, preventing pathogens from adhering. Next, no food. They consume available nutrients, starving potential invaders. Finally, chemical warfare. They produce acids and bacteriocins that directly kill or inhibit pathogens. Now, let us understand the importance of commensalism in health. First, healthy balance. A stable commensal population supports immune education and produces beneficial metabolites. If this balance is disturbed, it leads to dysbiosis. Dysbiosis can cause pathogen overgrowth, chronic inflammation, and loss of microbial diversity. Maintaining homeostasis is therefore essential for good health. Here, you can see a visual summary of commensalism. It begins with host-microbe relationships leading to commensalism, followed by colonization. Colonization provides benefits like protection and nutrition, ultimately maintaining health and homeostasis. With this understanding, we have completed this topic.